Hello and welcome to Humanized Immune System Mice for NK and T cells, applications for cell therapies and immune modulators, presented by Dr. Jessica Ramadhin. Let me now introduce our speaker. Dr. Jessica Ramadhin received her BA in Biological Sciences from Cornell University and earned her Ph.D. in Infectious Diseases from the University of Georgia. During her graduate studies, she tested 2 anti-inflammatory compounds as therapeutics for obesity and metabolic syndrome. She then completed her postdoctoral training in the Department of Gastroenterology at Cedars-Sinai Medical Center, where she examined how myeloid-specific knockout of the glycolytic enzyme hexokinase 2 impacted rodent models of obesity, colitis, and infection. Jessica has worked with animal models for many years and has a wide range of in vivo experience related to infectious diseases, immunology, metabolism, and the gut microbiome. We are delighted to have her with us today to discuss his mice for NK and T cells. So let's get started. Jessica, please begin when you're ready.
Good morning from Los Angeles. Thank you everyone for joining us. And as Dr. McBride mentioned, my name is Jessica Ramadhin and I'm a field application scientist here at Taconic. As a scientist, one of my roles is to provide technical and scientific support pertaining to Taconic's animal models. So I'm going to go ahead and turn off my camera for the remainder of this presentation, but we will be back for the Q&A portion. So first, I'd like to provide an overview of what I will cover in this seminar. I'll provide an overview of cellular immunotherapies and their importance to immuno-oncology research. I'll describe the most popular types of T cell therapies and how the HIL-2 NOG supports investigation of these therapeutic modalities. Next, I'll discuss emerging NK cell therapies and how the HIL-15 NOG supports these studies. And lastly, I'll provide a review of Taconic's NOG portfolio with a teaser or two about our newest models. First, I want to provide a little bit of background about cell therapies. So cell therapies represent an exciting advancement in the fight against cancer. And to date, there are 6 FDA-approved CAR T cell therapies, which are shown in the table here on the right. However, all approved therapies currently target blood cancers, as shown in the purple box here. And this is mainly because solid tumors present unique and complex challenges for treatment. Some of these challenges include tonic signaling, toxicity, mostly due to off-target effects or cytokine release syndrome, antigen loss, where the tumor downregulates the antigen, issues with trafficking or difficulty homing to the site of the tumor, activation-induced cell death, and exhaustion, which occurs when T cells lose effector function and upregulate proteins, such as PD-1 or CTLA-4. As I mentioned, there are 6 FDA-approved CAR T cell therapies available on the market, and CAR T cells continue to dominate the cell therapy pipeline. As shown in the graphic here on the right, there has been a substantial increase in CAR T cell therapies in various stages of development between 2020 and 2022. Other T cell therapies are also of interest, including TCR T cells and tumor-infiltrating lymphocytes, or TILs. NK-based therapies have also garnered significant attention, increasing 65% from 2020 to 2021 and 55% from 2021 to 2022. And based on these statistics, it's clear that cell therapy continues to be an attractive strategy for tackling cancer. So the need for relevant animal models is a huge area of importance. So TIL and TCR therapies are common T cell therapies that often involve ex vivo identification of T cells with an anti-tumor response. They're often autologous or isolated from the same patient who will be treated. For example, TILs are isolated from tumor biopsies and are already likely to react against tumor antigens. On the other hand, TCRs are isolated from the blood and genetically engineered to express anti-tumor receptors. These cells can then be expanded to large numbers for subsequent infusion into the patient and they can also be activated ex vivo to surpass the hostile tumor microenvironment. Another advantage to TIL and TCR therapies is that patients can be preconditioned prior to treatment in order to create a more favorable environment for T-cell expansion, persistence, and activity. So moving on to CAR T cells express synthetic receptors that target specific tumor antigens. And there have been several generations of CAR T cells, each increasing in complexity. First generation CAR T cells are considered the original CAR T construct. And second and third generation CAR T cells involve the addition of co-stimulatory domains to enhance T cell proliferation, cytotoxicity, and survival. Fourth generation CAR T cells involve the addition of inducible transgenic proteins such as IL-12, and these help enhance anti-tumor activity. Lastly, fifth generation CAR T cells include transcription factor binding motifs to reactivate the immune system and help maintain persistence. Various modifications have also been introduced into CAR T cells to alter function. This includes blockage of PD-1, alterations in scFv affinity, integration of suicide genes such as ICAS9, or incorporation of tandem CARs with multiple scFv domains to recognize multiple antigens. Novel methodologies are also in the works, such as epigenetic modifications, CARs that are induced by hypoxia, and ongoing discovery of future targets. So this brings me to introduce our HIL-2 NOG, which helps support investigation of T-cell immunotherapies. The HIL-2 NOG is a super immunodeficient NOG mouse expressing human IL-2. So why is IL-2 important? Well, IL-2 has a plethora of functions, but it most notably promotes T-cell growth and survival, as well as amplifies NK cell cytolytic activity. And in this regard, expression of IL-2 makes this an improved model for differentiation and engraftment of IL-2 dependent cells. Furthermore, this model eliminates the need for exogenous IL-2 support when assessing cell therapies. And when these models are engrafted with CD34 positive HSCs, the HIL-2 NOG survives up to 60 days and may be appropriate for some studies. In this model, HSC differentiation is heavily skewed to NK cells. However, we find that most customers interested in NK cells choose to work with the HIL-15 NOG, which I will cover later in this presentation. Lastly, we do not advise PBMC engraftment in this model due to very limited lifespan. And in the next set of slides, I will reference case studies and hopefully will convince you that the HIL-2 NOG is an optimal host for T-cell based therapies. Prior to development of the cytokine transgenic NOG strains, administration of exogenous cytokines was evaluated as a potential option. And as I mentioned, the HIL-2 NOG produces human IL-2 at a steady state. If we direct our attention to the figure here on the left, we can see that subcutaneous injection of human IL-2 into a base NOG mouse results in a peak 2 hours post-injection, but it is essentially gone within 6 hours. During studies involving cell therapies, it is not uncommon to inject IL-2 multiple times to help extend T-cell survival, and this can be laborious and stressful to an animal. If we look at the graph in the middle here with the red dots, we can see that the HIL-2 NOG consistently produces IL-2 at an average concentration similar to that of an injected animal. And based on user feedback, the cytokine levels in the HIL-2 NOG are sufficient to support engraftment of primary human T cells. In the figure shown here on the right, we can also see that this concentration remains stable over a period of weeks. There's a slight decrease in response to engraftment with CD34 positive HSCs, but this would be expected given that these differentiating cells will help utilize available IL-2. The HIL-2 NOG provides a solution here, in which this animal model eliminates the need for exogenous administration of cytokines. And to further emphasize this point, HIL-2 is also present in the tissues. In this slide, we are looking at IL-2 concentration in the spleen, liver, lung, and kidney. And these tissues contain a significant amount of IL-2 overall. However, IL-2 production in the spleen and liver is significantly reduced by irradiation alone. The HIL-2 NOG also produces lower amounts of IL-2 in several tissues after HSC transfer. Regardless, HIL-2 is present in both the plasma and tissues of this model to help support your adoptive cell therapies. Now, I'd like us to turn our attention to various case studies involving the HIL-2 NOG. In this first paper, Jesperson and colleagues expanded tumor-infiltrating lymphocytes from a patient with metastatic melanoma and labeled them with luciferase prior to injection. Next, they treated HIL-2 NOG mice expressing either low or high levels of IL-2 with these isolated TILs and found that the TILs survived the longest in high-expressing HIL-2 NOGs, as indicated by the blue line in the graph on the left. The low-expressing HIL-2 NOGs still supported TIL survival longer than the base NOG alone, as shown in orange. The figure on the right here shows bioluminescent images of these mice and persistence of the TILs from 1 day to 6 weeks post-injection. And as you can see, luminescence increased over time, and the high-expressing HIL-2 NOG exhibited the greatest level of bioluminescence at 6 weeks, thereby indicating T-cell proliferation and expansion. In this next study, we are looking at a patient-derived xenograft model of melanoma using the HIL-2 NOG. The pie chart on the top left represents an immunophenotyping of the TILs isolated and expanded from the melanoma patient. And as you can see here, most of these TILs were comprised of CD4 positive T cells. These same patient's tumor cells were injected into the NOG and the HIL-2 NOG, followed by infusion with TILs 5 days later. The TILs suppressed tumor growth in the HIL-2 NOG, as indicated by the flat green line, which you can barely see here, but not in the NOG mouse, as indicated by the blue line. On the bottom left, circulating T cells were measured in both the NOG and the HIL-2 NOG mice at several time points post-infusion. And while there are virtually no circulating T cells in the NOG, it's quite striking that the circulating T cells persisted until completion of the study in the HIL-2 NOG. Lastly, the researchers injected cryopreserved biopsy cells to determine if the TILs could be reactivated without ex vivo culture. This was the case as tumor growth was still suppressed in the HIL-2 NOG using these cryopreserved cells, thus bypassing the need for expansion. Here, we are looking at another PDX model of melanoma, where the mice were treated with TILs plus pembrolizumab, which is an immune checkpoint inhibitor that blocks PD-1 on T cells. And as indicated on the graph in the graph on the left, HIL-2 NOG mice treated with both TILs and pembro experienced complete regression of tumor growth. This is depicted here by the green lines. Control NOGs treated with TILs and pembro, as well as HIL-2 NOGs given pembro alone, did not exhibit the same tumor regression. This is further evidenced by the bioluminescent images shown here on the right, where the two control groups have significant bioluminescent signals, and the signal is virtually non-existent in the HIL-2 NOG mice treated with combination therapy. In another PDX model of melanoma, researchers wanted to test whether caronitinib would interfere with T-cell mediated anti-tumor immunity. Caronitinib is a drug that inhibits nucleotide sanitizing enzyme and has been shown to have a good cytotoxic effect in cancer cells. In the graph on the left, HIL-2 NOG mice were injected with tumor cells and treated with either the vehicle caronitinib TILs, or the combined treatment. Carotid did not impair TILs as evidenced by the nearly identical tumor growth with or without the drug. The image on the right shows representative bioluminescent images of the tumors from the mice, showing effectiveness of the TILs and the combined treatment method. At this point, hopefully I've convinced you that the HIL2 is beneficial for assessing TILs, but what about TCR transduced T cells? In this paper, Arnaud et al. Used a novel platform called NeoScreen to identify T cells specific for neo or rare tumor antigens. Then they generated neoantigen-specific TCRs, which were injected into the HIL2 mice 2 weeks post-engraftment with melanoma tumor cells and monitored over 40 days, as shown in the timeline here. The graph on the right shows that treatment with these TCR transduced T cells resulted in an inhibition of tumor growth, as denoted by the orange line. Here is another example of TCR T cells being tested in the HIL2 in a cell line-derived xenograft model of B-cell lymphoma. In this model, a B-cell lymphoma cell line was injected into both NOG and HIL2 mice. And after tumors reached a certain size, mice were treated with a single injection of mock T cells or TCR T cells. The figure on the top left here shows tumor volume and % survival in the NOG mice. And while tumor growth was inhibited in the beginning, there was a relapse in the TCR T treated animals. There was no relapse during these experiments in the HIL2 NOG, as shown here on the right. And the flow cytometry plots on the bottom left show that these TCR T cells persisted in the blood, bone marrow, and spleen of the HIL2 NOG compared to the base NOG. Lastly, the TCR T cells in the HIL2 NOG did not exhibit a significant increase in exhaustion. And this is shown here, where we're looking at increased PD-1 expression in the tumor of the base NOG, but no signs of exhaustion in the blood, bone marrow, and spleen. In this next study, the researchers engineered T cells with specificity for Wilms tumor 1, a transcription factor overexpressed by a wide range of tumors. The timeline here shows that NOG, HIL2 NOG, and HIL15 NOG mice were injected with leukemia cells, followed by TCR T cells after 2 days. Animals were followed until the endpoint at day 27. And I know we haven't gotten to the IL-15 NOG yet, but here's a sneak peek at one of its applications. The HIL2 NOG and HIL15 NOG mice treated with Wilms tumor 1 TCR T cells had significant infiltration of cells in the bone marrow, as shown here on the top right. Tumor progression was also slowed in the TCR T cell treated NOG models, but the HIL2 and HIL15 NOG still showed the greatest effects due to increased support for the cell therapy. We've discussed TILs and TCR T cells so far, but what about CAR T cells? This study is exciting because several different CDX and PDX models of melanoma were engrafted into the HIL2 NOG to evaluate HER2 CAR T cells. First, the figures shown here on the top of the slide depict patient-derived xenografts of melanoma known to either be resistant or regress in response to adoptive cell therapy. HER2 T cells caused nearly complete regression of all tumors in the HIL2 NOG, as denoted by the peach lines, but not in the control NOGs shown in green. The bottom left graph uses a commercially available cell line of uveal melanoma and also shows tumor regression in the HIL2 NOG after CAR T cell therapy. And lastly, the researchers established a novel PDX model of metastatic uveal melanoma, wherein the treated HIL2 NOG mice also exhibited tumor inhibition. I'm not showing it here, but they attribute this to the presence of CD3 positive T cells in the regressing tumors and blood of the HIL2 NOG mice. And lastly, there's a nice quote in this paper, as shown here on the right, pointing out that the HIL2 NOG mouse is the optimal humanized mouse model to test novel CAR T cells against solid tumors. So, does what we observe in the HIL2 NOG translate to the clinic? The figure on the top left of this slide depicts monitoring of disease progression via measurement of plasma S100B in a patient. The corresponding timeline below the graph shows how the patient was treated and when surgical removal of subcutaneous metastases occurred. These surgical biopsies were then engrafted into the NOG and HIL2 NOG at different time points. The biopsy shown in the first graph here on the bottom left represents the patient's tumor prior to treatment. And as you can see, the tumor cells grew aggressively in both models and corresponded to the extensive metastases seen in the patient on the top right at day 23. This particular biopsy was composed of highly glycolytic tumor cells and a few TILs, so the recommended treatment was dabrafenib and trametinib for this patient. The next biopsy, which was taken after treatment with dabrafenib and trametinib, did not grow at all in the NOG and HIL2 NOG and corresponds to a significantly lower level of metastases seen in the patient at day 55. This particular biopsy contained mostly TILs, which prompted doctors to switch this patient to anti-PD-1 therapy or pembrolizumab. Tumor burden decreased in response to pembro, but new lesions appeared that again grew in the NOG and HIL2 NOG, as shown in the third graph in the bottom row. And lastly, new metastases occurred in the patient, and this was resistant to immunotherapy in both mouse models, as shown in the fourth graph. And this prompted a switch back to dabrafenib and trametinib due to the robust response that was seen initially. And while this case study is complex, it really emphasized that response of the PDX to treatment in the HIL2 NOG correlates to clinical outcomes. In the study shown here, 6 different tumors and TILs were isolated from patients. MM11, 5, and 24 were responders, while MM29, 46, and 4 were non-responders in the clinic. The graphs shown in a column on the left depict the responders to TIL therapy. And as you can see, the TILs from these patients inhibited tumor growth in the HIL2 NOG, as shown in black. On the other hand, the graphs shown in a column on the right depict non-responders to TIL therapy. The TILs here were ineffective at preventing tumor growth in the HIL2 NOG, similar to what was seen in these patients. So we've talked a lot about immuno-oncology thus far, but what about other applications for this model? This paper was just published in PLOS One, establishing a xenograft model of psoriasis using the HIL2 NOG. In this study, xenografts of psoriatic lesions were engrafted in CB17 SCID, NOG, and HIL2 NOG mice. And as you can see in the histology images below, T-cell infiltration occurred most abundantly in xenografts of the HIL2 NOG. T-cell infiltration was also seen in the lymph nodes and blood, and the researchers also observed higher cytokine levels in the graft lysates and serum. This included IL-17A, IL-22, interferon gamma, and TNF-alpha. The caveat of this model, however, was onset of graft versus host disease, beginning at around 20 days after surgery, and thereby significantly limited the time window for this study. This paper represents an interesting step towards developing autoimmune disease models in the HIL2 NOG, and we expect much more of this in the future. As we saw in the previous slide, the HIL2 NOG exhibits limited lifespan, and for this reason, we tend to recommend that these mice be used for studies involving adoptive cell therapy instead of full humanized immune system reconstitution. As you can see on the figures on the left, HSC reconstitution in the HIL2 NOG results in a high percentage of human CD445 positive cells in the blood, but this population is heavily skewed toward NK cells. And as noted in the psoriasis study, these mice succumb shortly after engraftment. In the case shown here, female HIL2 NOG mice survive for 40 to 60 days after HSC engraftment. Limited lifespan is also observed in the HIL15 NOG, which I will discuss later on in this presentation. And when engrafted with PBMCs, the HIL2 NOG survives for less than 2 weeks. And in light of this data, it's best to consult with one of Taconic's field application scientists to help determine which humanized immune system model is most suitable for your studies. So I've hinted quite a bit at the HIL15 NOG, but I'd now like to discuss this model in more detail and provide case studies about how it can help support investigation of NK cell immunotherapies. The HIL15 NOG is a super immunodeficient NOG mouse expressing human IL-15. Why is IL-15 important here? Well, while IL-15 has numerous functions, it is most notable for promoting NK cell growth, survival, and cytotoxicity. And in this regard, expression of human IL-15 makes the HIL15 NOG an optimized model for engraftment of isolated primary human NK cells, immortalized human NK cell lines, or human NK cell therapies. We do not advise PBMC or HSC engraftment in this model due to limited lifespan post-engraftment, and I will discuss this in later slides. So a little bit about NK cell therapies. These types of therapies are rising in popularity, and there are currently 3 strategies for this type of treatment. In autologous NK cell transfer, NK cells are isolated from a patient, expanded ex vivo with cytokine support, and then reinfused. Historically, IL-2 has been used for expansion, but it is now known that combinations of IL-12, IL-15, and IL-18 can also be used. In allogeneic NK cell transfer, donor NK cells isolated from either HLA-matched or partially matched donors are expanded and infused into a patient. And lastly, CAR-engineered NK cells can be generated from both autologous or allogeneic NK cells, as well as from NK cell lines. And similar to CAR T cells, CAR NKs can have multiple co-stimulatory domains, altered antigen specificity, etc. Compared to T cells, NK cell therapies have unique advantages. They're attractive for adoptive cell therapies because, unlike T cells, NK cells are not tailored to specific antigens and tend to attack abnormal cells. They are also generally not associated with graft versus host disease, and they can be acquired from different sources, such as cord blood, PBMCs, and even immortalized cell lines. I won't go into it in detail, but the chart that I'm now showing on the right gives you a variety of examples of NK cell sources, as well as their specific advantages and limitations for your reference. As with the HIL2 NOG, the advantage of the HIL15 NOG is that it constitutively expresses a human cytokine and avoids the need for exogenous administration. And as shown in this plot here, HIL15 is found in a variety of tissues in this model, such as the spleen, liver, lung, etc. In the original HIL15 publication from Catano et al., NK cells were isolated from PBMCs and engrafted into this model and monitored over a period of 24 weeks. If we look at the graph on the top left, we can see that the frequency of NK cells increased for 2 weeks post-transplantation and gradually decreased for 6 weeks thereafter. In the NOG mice, NK cells disappeared within 10 to 14 days. On the bottom of this slide, we can also see that the absolute number of NK cells peaked around 4 to 5 weeks, and the flow plots shown here on the right demonstrate that these NK cells migrated to the spleen, liver, and lung, and some are also found in the bone marrow. These cells also maintain expression of human CD16 and HuNK P46, as well as expression of CD56. It is also important to note that these NK cells persisted without graft versus host disease. And to elaborate on the previous slide, the engrafted NK cells isolated from the HIL-15 NOG remained functional. On the top left, we can see that expression of NKG2 family members remained fairly consistent between fresh, as shown in white, and isolated NK cells, shown in black. In fact, 2 of these receptors were actually significantly higher in isolated NK cells. And these are NKG2A and NKG2C. On the bottom left, we see no differences in killer immunoglobulin-like receptors or in CD57 or CD8 maturation markers. On the top right, we also see that there is no difference in levels of TBET and EOMs, which are crucial transcription factors for NK cells. And lastly, I'd like to point out the high levels of perforin in the plasma. Perforin A is a protein released from NK cells and indicative of cytolytic activity. Overall, this slide demonstrates that the isolated NK cells from the HIL-15 NOG mice retain their immunological features. In this poster presented at ASGCT in 2020, HIL-15 NOGs were injected with primary NK cells followed by injection of K562 tumor cells 4 weeks later. And if we look at the imaging on the left at 3 hours post-tumor injection, we can see that the NK cell treated mice have virtually no radiance, indicating limited outgrowth of the tumor. The imaging on the right shows spread of the tumor throughout the control mice versus no tumor growth in the animals administered primary NK cells. This further exemplifies the retained functionality of primary NK cells in vivo in the HIL-15 NOG. Now, I'd like to direct your attention to case studies involving CAR NK cells. In this particular study, CAR NK cells targeting CD123 were designed and tested in a CDX model of acute myeloid leukemia. The experimental timeline is shown on the top left, wherein the mice were injected with cells 3 days prior to infusion with CAR NK cells. Animals were maintained on this study until the endpoint at day 50. And as we can see on the bottom left, tumor growth was suppressed in the HIL-15 NOG mice that were administered the CAR NK cells. This is also exemplified in the imaging here, where the animals treated with CAR NK cells exhibited very little bioluminescence compared to controls. Using the same experimental design I presented in the last slide, the researchers measured various cytokine levels in both the HIL-15 NOG and the NSG. In the case of the HIL-15 NOG, they were able to detect anti-leukemia effects, expansion of effector cells, and cytokine secretion in this model. This included elevations in TNF-alpha at day 15, as well as granzyme B and interferon gamma at day 30 in CAR NK cell treated animals. However, despite anti-leukemia effects, they were unable to detect effector cells and cytokine secretion in the NSG model. And this is shown here in the figure on the right. Now, the study shown here tested induced pluripotent stem cell-derived CAR NK cells in a CDX model of mantle cell lymphoma using the HIL-15 NOG. In this set of experiments, HIL-15 NOGs were engrafted with GECO1 cells 4 days prior to infusion with CAR NKs. And after infusion, animals were injected with IL-2 every 3 to 4 days, and images were taken at various time points. And as you can see in the imaging shown here below, these CAR INKs had variable levels of efficacy at day 12. And this particular CAR INK, PCB7142, was the most effective. Next, I'd like to discuss 2 examples of antibody-dependent cell-mediated cytotoxicity activity in the HIL-15 NOG. In the experiment shown on the left, mice were inoculated with a human gastric cancer cell line expressing HER2. Mice were then engrafted with NK cells, plus or minus an anti-HER2 antibody, Herceptin. Tumor growth was suppressed in the group that received combination therapy. In the second example, shown here on the right, a CCR4 positive Hodgkin's lymphoma cell line was used. These mice were treated with NK cells plus or minus an anti-CCR4 antibody. And again, the mice treated with a combination showed suppression of tumor growth. Again, I want to touch on lifespan limitations in this model with certain engraftment methods. The HIL-15 NOG can be engrafted with HSCs or PBMCs, but survival is limited. When reconstituted with HSCs, HIL-15 NOG mice die within 40 to 70 days post-engraftment, depending on the number of cells injected. This is shown in the graph on the left, where HIL-15 NOGs engrafted with 25,000 HSCs, for example, succumb at 50 days post-engraftment, indicated by the green line. When reconstituted with PBMCs, as shown on the right, HIL-15 NOGs succumb within 30 to 40 days. And while this model may not be ideal for humanization with HSCs or PBMCs for long-term studies, it is useful for studying actual graft versus host disease and other adverse outcomes. Furthermore, we are not aware of lifespan limitations with purified NK cell engraftment, and this does not seem to be a limiting factor for those types of experiments. So beyond cancer in the HIL-15 NOG, I'm showing a study here from Insight, where 3 different concentrations of PBMCs were engrafted into the HIL-15 NOG to study graft versus host disease. The chimerism is shown in the top figures, with increasing percentages of HCD45 correlating with the number of engrafted cells. These HSCs differentiated into CD3 positive cells and CD56 cells, as shown in the 3 bar graphs on the top right. These mice exhibited rapid onset of graft versus host disease, with weight loss beginning around 12 days post-engraftment. Disease severity also increased, as indicated by graft versus host disease score, and survival decreased, with humane endpoints being reached within a month. At sacrifice, the researchers noted significant PBMC infiltration in the GI tract, lung, liver, skin, eye, and pancreas. This is more widespread than what has been seen in traditional humanized preclinical models of graft versus host disease. In the same study where the graft versus host disease model was established, the researchers decided to test the efficacy of ruxolitinib, which is a selective JAK1/2 inhibitor, during graft versus host disease. The figure on the left shows graft versus host disease score over time, where treatment with the vehicle prednisolone, prednisolone plus ruxolitinib, or ruxolitinib alone began at 10 days post-engraftment. Ruxolitinib ameliorated acute graft versus host disease and lessened weight loss, as shown in the graph here in the middle. Therapeutic treatment with ruxolitinib also increased survival, as shown here on the right. And this provided proof of concept that these animals can be used to study drugs against graft versus host disease, which is a common concern for patients receiving cell therapies. In the second example of graft versus host disease, Bionis Therapeutics tested a dual-specific IL-15, IL-21 inhibitor to ameliorate intestinal graft versus host disease. In the histology shown on the left, we see tissue damage and immune infiltration in the lamina propria of humanized IL-15 mice. Intestinal permeability was also significantly increased, as evidenced by the leakage of phytidextran into the blood following oral gavage. And after providing evidence of intestinal graft versus host disease shown here, the researchers treated these mice with either vehicle, BNZ2, or anti-IL-15 antibody. And the endpoint histology is shown here on the bottom right, where BNZ2 and anti-IL-15 treatment significantly reduced HCD45, CD8, and CD4 T cell infiltration into the tissue, as well as prevented disruption to the gut barrier. This effect was specific to the gut, as cell engraftment was still observed in the blood and the spleen. So I'd like to provide a summary of what we've covered and considerations for using humanized immune system mice. Here is a high-level overview of the models covered, and as you can see here, each model supports specific immune cells based on the available cytokine support. Well, how do you choose an appropriate model? Choosing a HIS model can depend on a variety of factors. In our first webinar, Dr. Mayara Lake introduced the superimmunodeficient NOG mouse, which can support PBMC and HSC engraftment and provides superiority in terms of difficult to engraft tumors. The NOG-EXL supports a more complete humanized immune system with differentiation of myeloid lineages. It is useful for a variety of applications extending beyond immuno-oncology, and it is readily available as an off-the-shelf humanized model as the HuNOG-EXL. The HIL-6 NOG can also be used for studies targeting myeloid and lymphoid cells. And lastly, I just covered the HIL-2 and HIL-15 NOG. The HIL-2 NOG is useful for assessing T-cell immunotherapies, but it can also be used to study NK cells. The HIL-15 NOG has been used to study T-cell therapies, but it really excelled for NK cell therapies. And these models exhibit limited lifespan when engrafted with HSCs or PBMCs, so they are best utilized for evaluation of adoptive cell therapies. Not only are there a variety of models to choose from, there are also a number of special considerations when using these models. So first, what type of humanization is required? Do you need HSCs? Do you need PBMCs? Or a specific cell type? What is your cell type of interest? And is it present after humanization? Are you looking to onboard a cell line or a patient-derived tumor? And how long is your study timeline? There are slow versus fast-growing tumors and short versus long treatments. Next, how readily available are these models? Do you need a HuNOG-EXL off the shelf, or do you require specialized engraftment? Variability is also a concern when using donors. How many mice do you need per donor, and how many donors need to be represented in a study? Lastly, there are also considerations for animal housing and husbandry, and we're here to help. So please don't hesitate to schedule a consultation with a field application scientist to help design your study. And with that, thank you for your attention, and I will now be taking questions.
Thank you, Jessica, for that fascinating presentation. Let's start with our first question. You talked extensively about cell therapies, but you did not spend as much time on novel immunotherapy drugs. So are these models suitable for use in studying immune-engaging molecules?
Yeah, so I focused on cell therapies because it's such an exciting and high growth area. And as I showed in the statistics graph, right, it continues to increase. But many scientists are using these models to study immune engaging drugs, such as bikes and trikes. And the HIL-15 NOG mouse has been used to study various NK cell engaging molecules in combination with primary NK cells or NK cell therapy engraftment.
Our next question is about the IL-2 NOG. So you showed that it had been used to develop a xenograft model of psoriasis. What other applications does the HIL-2 NOG have outside of the realm of immuno-oncology?
That's a great question. So while our customers are doing very interesting work, the xenograft model of psoriasis is, the first published instance of autoimmune disease in the HIL-2 NOG. If you have additional ideas about utilization for the IL-2 NOG and want to discuss potential collaborations, especially in the space for autoimmune disease, we'd be very interested in that. So please reach out if you'd like to test a new use for this model.
Our next question is about NK cell therapies. These are attractive because they can be derived from different sources. So can you speak a little bit in more detail about the sources of NK cells and some of the advantages and limitations of those?
Yeah, so NK cells, as I mentioned, are attractive, right? Yeah, like you said. So they can be derived from different sources. They don't have to be sourced directly from a patient. So they can be expanded from cord blood, HSCs, peripheral blood, and even immortalized cell lines. And yeah, there are a variety of advantages and limitations to this. So for example, cord blood is attractive because it's readily available. But it is subject to donor variability, and it does require a few weeks of expansion ex vivo. HSCs are another source available via, cord blood banks, and they contain NK cell precursors. They require a long expansion time and are also subject to donor variability. And another caveat of using HSCs is they exhibit low antibody-dependent cellular cytotoxicity because they have low expression of CD16. On the other hand, if you use peripheral blood, these cells isolated from peripheral blood are mature and highly cytotoxic. However, using peripheral blood is not readily available and requires donors. And again, the issue of donor variability arises. And lastly, if you want to use immortalized cell lines, I think the NK92 is most commonly used. Key advantages here, it's a homogeneous product and it's easy to engineer for whatever your target is. And the disadvantage here is that these cells need to be irradiated prior to infusion because they are of NK cell lymphoma origin. They also don't last as long because of this irradiation, and similar to HSCs, they also have low ADCC due to limited CD16 expression.
Oh, a lot of things to consider there when selecting a cell source.
Yeah, absolutely.
So most of the examples that you showed used either the HIL2 or the HIL15 NOG, but one of the studies used both of the models, and that was for a TCR-T cell therapy. Could you speak to what the value of using both of those in an experiment might be?
Yeah, so as cytokines, IL-2 and IL-15, they have, tons of pleiotropic functions, but they both have similar properties related to T cell survival and proliferation. One of the caveats of IL-2, however, is that it can play a role in activation-induced cell death of T cells, which I mentioned as one of the challenges involved in developing effective cell therapies. IL-15, on the other hand, might be advantageous here because it might help limit toxicity. And another, disadvantage to using IL-2, it can also hinder memory CD8 T cells, while IL-15 is known to maintain these cells. So yeah, it just depends on, the function of these different cytokines.
I want to thank you again, Jessica, for that fascinating presentation.